we can move right along to our main presentation for today. So let me introduce our featured speakers, which we have two of today. Our first featured speaker will be Rob Budd, a research scientist with the Environmental Monitoring Branch within California's Department of Pesticide Regulation, or DPR. Rob received his PhD in environmental science from the University of California at Riverside and has worked at DPR since 2009. His primary duties include serving as a lead scientist, coordinating monitoring projects intended to determine the status and trends of pesticide concentrations within California surface waters. His interests include evaluating the ecological relevance of pesticide loading within watersheds and developing mitigation strategies to reduce the deleterious effects on aquatic ecosystems. Rob will be delivering a longer presentation to our group this morning, and then we'll be hearing a shorter presentation from our second featured speaker, Autumn Ross, who is the pollution prevention specialist in the Wastewater Enterprise of the San Francisco Public Utilities Commission, SFPUC. Autumn has a background in estuarine ecology, including restoration of native eelgrass beds in the San Francisco estuary. And at SFPUC, Autumn now protects water quality by helping to prevent pollutants at their source, thereby reducing the amount of pollutants entering the sewer system. And so what we'll hear about today is how DPR's Urban Surface Water Monitoring Program and PUC's Wastewater Enterprise track pesticides, sample and analyze them, share findings, and apply regulatory context to guide IPM practices. And I'll just add that I'm really excited to hear from Rob and Autumn about their pesticide monitoring work today. Here in our audience, we typically have a lot of IPM practitioners, and so you may be applying pesticides as part of your routine jobs. And whatever pesticide products you're using, they're getting not only to your intended target, but some amount is going to move to soil, air, and water. So once the product's applied, it's in our environment. And I think that monitoring programs like the ones that Rob and Autumn will speak about are really important for tracking pesticide levels and informing our IPM practices and policies. So I'll pass it over to Rob and let you take it from here.
Okay, thank you very much. Can you hear me okay?
Yep, can hear you great.
Great. Let me just, it'll just take me a second to set up. And tell me when can you see my slides okay?
Yeah, see the slides, They look good.
Awesome, okay. And as I mentioned, if there's questions or anything, just let me know because I won't be able to necessarily see them as I present. But yeah, good morning, everyone. Thank you for joining and thank you for inviting me here to talk. As mentioned, my name's Robert Budd. I am a research scientist with DPR's Surface Water Monitoring Program since 2009 to date me a little bit. Yeah, and today I'll be talking about our urban monitoring project, specifically looking at pesticides that move off the urban environment and into the local waterways. And currently my job is to sort of coordinate all the monitoring projects, but I still do get out in the field. My first job was part of our Southern California monitoring team. And I still get out there and I've taken these pictures at a couple of our local sites down there. Let's see. Okay, so the first thing I'm going to do is just to give you a brief overview of our program and individual projects. For those of you that are not as familiar with DPR and our workings and what we're all about, this is a lineup of all the branches within DPR. We've expanded quite a bit since I started. We now have branches for registration, evaluation, human health, worker health and safety, enforcement, IPM, and environmental monitoring. And within the environmental monitoring branch, we have 3 programs that monitor for pesticides in different environmental matrices, including air, groundwater, and surface water. And as I've mentioned, I work for the Surface Water Protection Program. And all of the branches work together to try to fulfill DPR's mission of protecting human health and the environment by regulating pesticide sales and use and by fostering reduced risk pest management. That's a new, a lot of words, but it means a lot to what we do. And yeah, and for those of you, I know that this is an IPM group, so this is a group that you might want to reach out to because they have a lot of great resources at their disposal about IPM practices of pesticide applications. Okay, so we're responsible for protecting California's surface water by ensuring that the concentrations of pesticides don't reach levels that are potentially toxic to aquatic habitats. And this includes fish and invertebrate communities, as well as plant species. Part of this process is to identify sources of pesticides that are reaching our surface waters. So we have developed programs to measure pesticides in agricultural and urban runoff. And new to our program is to measure wastewater effluent at some volunteer wastewater facilities. But for today's presentation, I'm really going to be focusing on the potential for pesticides to move off the urban landscapes through irrigation and rainfall into the storm drains and then finally into our receiving waters. So the next couple slides, I'm going to be talking about our sampling design. The first question I will talk about is where do we sample? We've split our monitoring locations into storm drains and receiving waters. So storm drains are important because they're a very important conduit, moving excess water off the urban landscape into the receiving waters. We generally use the data that are generated from our storm drains for source identification to identify regions of high pesticide use and what's coming off the landscape, and to also to link to land use data, which includes in the urban environment, industrial, commercial, and residential land use types to try to track back what are the important land types that are contributing to pesticide loading in our waterways. Receiving waters, on the other hand, are where our aquatic habitat that we're trying to protect resides. So these are more ecologically relevant waterways or waters that we usually compare the concentrations to toxicity thresholds to figure out if there is a potential concern of runoff. And yeah, they're usually a larger contributing area than the storm drains are like a microcosm. So here is a picture of our two figures showing our current monitoring locations throughout the state. The waterways that I just talked about are represented by the green squares, and the storm drains are represented by the red circles here. And we split the projects into Northern and Southern California. And as you can imagine, we really focus on the major urban hubs throughout the state. In Northern California, we have locations in the Sacramento region and in the Bay. And in Southern California, we have a lot of locations in Los Angeles, Orange County, and San Diego counties. And since this is a San Francisco group, I've just put a circle around our Bay Area monitoring locations here. So another important question about our monitoring is when do we sample? So we also split our monitoring scheme into two different seasons. The dry season, which is generally during the summer, between June and September. They're usually characterized by lower flows, and it's really water that from over irrigation, right? From over watering lawns and washing cars. I'm always amazed by, I was in charge of the Southern California project for many years, and they would typically not get any water for 4 or 5 months, and but I never had a time when I couldn't collect runoff from the storm drains because there's always a lot of water from just over irrigation. And then the other side is measuring runoff during rain events. And these are obviously higher flows. Rain events can be flashy, which we see down in Southern California, where you get just a little amount of rainfall and you get a big amount of runoff, or you have these long, prolonged events that are more characteristic of Northern California regions. And I just put these pictures up because I like to these as a comparison because they're taken at the same site 2 months apart. This on the left was during the dry season runoff, and you can see there's just very a little bit of trickle, and then after a very moderate amount of rainfall, it can really transform the site into this raging river. And you can imagine that everything from the surrounding watershed and urban landscape just flushes down into this storm drain, and sometimes we can't even get in to sample where we want to. And for the rain events, I will mention we try to get the first rain event of the season, which is called the first flush. Generally, a lot of monitoring programs like to do this because it's really where a lot of the contaminants, that have been built up over the dry season that haven't been washed off, it has the greatest potential to be carried during that first flush event. So it's really important that we try to capture some of that rainwater during that period. So another question is, how do we decide what and where to sample? We've developed an internal prioritization model to help us direct our sampling plans. On the left here is the user interface, which a user would put in the parameters of interest, including if they're looking at just agricultural use or urban use or both, and the toxicity thresholds of concern, and then also the pesticide use. This is very outdated. That we have much more current pesticide use than 2014, and we're currently, I think, at 2022. So all this, you put in the parameters, and then on the right, it generates an output. This is just a representative output of what you might find. And how the model works is all professional use in for agricultural and for urban landscapes by professional applicators is reported to DPR's pesticide use reporting system. And that would be represented for each individual chemical right here. And from that, the model generates a use score. It's a ranking, relative ranking use score. And then same for the toxicity. We generally use the minimum US EPA aquatic benchmark for toxicity thresholds. And this generates also a ranking score for toxicity. And the model then just multiplies those two together to give you a final score. So that the higher the score, the higher the priority for monitoring. And then lastly, the model will provide a recommendation for monitoring. So and this is based on the physicochemical properties of the chemical. And in this example, you can see that all of the chemicals are suggested for monitoring except for chlorothalonil, which has a very short persistence in the environment, so it's not believed to be as found as frequently. All right. Next, I'm gonna just dive right into some of the data that we've found over the years. And I apologize in advance. These are all busy bar graphs, so I'm going to try to take them slow for so you don't start nodding off. This first one, really, I like to start with. It represents all the data that we've collected at all site types and all event types. And I wanted to keep it current, so I just pulled out data from 2020 to 2025. Along the y-axis, and this will be the same for all the bar graph slides, you'll see the detection frequency, and then along the x-axis is the name of the pesticide of concern. The orange bars represent the detection, or how often we're detecting above the reporting limits of the chemical. And then the red bars are a percentage of the concentrations that are above the aquatic minimum benchmark. So this relationship shows a very important concept to our program, that about ecological relevance, because just because a chemical is detected doesn't necessarily mean we consider it an ecological concern. The great comparison is, for glyphosate, we're detecting it in almost every sample. Over like 84% of all samples across the state, we're detecting it. But the toxicity threshold is so high that the concentrations never break it. Conversely, imidacloprid and bifenthrin, which are both insecticides in common products, they're also detected at greater than 70% of all samples. And because their toxicity thresholds are so low, we're detecting them almost every single time we detect it, it's an exceedance. In fact, for bifenthrin, it's below the reporting limit. So if we detect it, it is an exceedance. So I also highlighted down here 3 important chemistries that you'll be hearing about throughout the talk. So the yellows represent pyrethroids, which are in many different urban use products, and those include bifenthrin, permethrin, deltamethrin. Basically, whenever you see a thrin at the end of a chemical, it's generally a pyrethroid. The fiprils, which represents fipronil, is the parent compound, and then 5 different degradation products are the fiprils. So these degradation products are not found actually in the products, but this is what it degrades to in the environment. So those are represented in purple, and the neonicotinoids, which have gotten a lot of publicity lately for their potential association with the bee colony and their effect on them. They're represented here in light blue. And as you can see by me just pulling these out, almost all the bars with associated red exceedances are associated with one of those 3 chemistries. So because this is a Bay Area meeting, I decided to just pull out all of our Bay Area monitoring data and do a little, have a little comparison. And same thing where I looked at all the sites between 2020 and 2025. And we see a similar pattern between this and the statewide, except I'll point out that we're detecting a lot of the higher priority chemicals at a lot less frequency than the rest of the state. So this is a good news for the Bay Area, where imidacloprid and bifenthrin are detected at probably half of the frequency as the rest of the state, which is really great news. Now I will put in a little caveat, if for those of you really paying attention, that the Bay Area currently only has receiving water site monitoring sites, and one of our currently this year, we're trying to add some storm drains so we can have a more apples to apples comparison with the, our sites in Sacramento, because as you'll see, that really comes into play with the differences in site types. We know that storm drains contribute, a lot to the overall picture of pesticide detections. And I throw this slide up as well because it is an important concept in looking at the event types comparisons. We look at, as I mentioned, we compare the storm season versus the dry season, and so the storm really is especially in the Bay Area, is contributing a lot of the potential detections. As you can see that it's storm runoff is more than double for most of the active ingredients detection frequencies between compared to the dry season. And this it really is evident for the Bay Area. When I've compared, I don't have the slide for the statewide comparison, but again, it's really distinct. Some of our Southern California sites, this would not be the case, where the dry season runoff actually carries quite a bit of fipronil and some of the pyrethroids. But in the Bay Area, it's very distinct that there's a lot less during the dry season, which is, good news, and it helps inform other monitoring programs in terms of their monitoring efforts, because you can really kind of focus in on the storm runoff as compared to dry season. So up to this point, I've been really talking about the theoretical toxicity or theoretical risk of different chemicals by using the comparison to the EPA benchmarks. So those are, the benchmarks are set by laboratory analysis, and then they do some math, and then they create these benchmarks. Now, we also do direct toxicity testing at a subset of our locations, where we use this creature up here. It's a little funny guy, Hyalella azteca, because they're very sensitive to pyrethroids in particular. So this is, I know this is also kind of a busy slide. So these black dots represent the point on the curve where on the y-axis you have the total pyrethroid concentration, and then the y-axis is the Hyalella azteca survival from 0 to 100%. So what you'd like to see, right, is everything up here at 100%, because that means everything's fine and dandy in your sample. But what we're noticing is that there's a distinct relationship between concentration and survival, with a dramatic an increase in pyrethroid concentration dramatically reduces the survivability of the Hyalella. And you can see that the relationship is pretty strong here. After a certain point, they're pretty much all dead. And what's interesting is when you pull out just bifenthrin concentrations, that it shows that it can, almost all the mortality can be explained by bifenthrin concentrations alone. So we know that this is a high priority chemical for us. And as I showed on the previous slides, we're detecting it at quite high frequency. So this is definitely on our radar for high priority chemical. And this is, again, showing a direct observed toxicity versus a theoretical, which, you can argue what, if those values are an apple to apple comparison with what we have in our waterways. But this is actually species that we're detecting we're observing from our waterways. Okay, so that's the end of the data. So now I'm going to shift to mitigation measures that we've implemented to help correct some of these or mitigate some of these concentrations in our waterways. The first is the surface water regulations, or we should have just named them the pyrethroid surface water regulations. They went into effect in 2012, and they apply to all professional applicators, including landscape maintenance gardeners. And it affects 17 different pyrethroids. And as I pointed out earlier, if it almost for the naming convention, almost all of them except for S-phenvalerate, if they have the thrin at the end, it's a pyrethroid. So you can look at your the next product that you use and see if that's there. And in general, what the goal of these regulations were to just reduce the amount of pyrethroids in runoff. So this figure depicts the allowable applications of any professional use of pyrethroid products for structural pest control. So it's a little busy, but it basically just breaks down to any application to the structure, like beyond the perimeter, or the hardscape, impervious surfaces, which includes the driveway and the sidewalk, and also a 2-foot buffer surrounding any of the hardscapes from the all around the driveway and all around the sidewalks. The applications are limited to a pin stream, a crack and crevice, or a spot treatment. And then around the perimeter, we've reduced the perimeter band to a 2-foot up from the ground up to the up the surface. And so these are, I'll point out, so regulations are, although they're not on the label, and they are often more restrictive than what the label allows, they're still enforceable by law, and they need to be followed by the professional applicators. Oh, and also that they do not allow any applications to any French drain or drain that leaves the property. So because I think I've highlighted this, that bifenthrin obviously has additional concerns from the other pyrethroids as being a major problem. So there's also a bifenthrin memorandum of agreement between the EPA and the registrants and DPR that it applies to all professional products containing bifenthrin used for structural outdoor applications. And it basically prohibits applications to the impervious surfaces, including the driveways and sidewalks. And you'll hear these themes regurgitate often because we know that the driveways and the sidewalks are the major conduit for transporting pesticides off the landscape and into our storm drains. You know, the water, things don't stick to the concrete as much, and it doesn't have a chance to soak in, like, you know, on the turf or ornamentals or gardens. There's at least a chance for the pesticides to be transported downward instead of directly off the landscape, whereas the driveways and sidewalks, it's just a straight shot. So we know, you know, from a lot of previous research, we know that they're the most important conduit. And so a lot of the mitigation strategies are set up to try to address this area. And it basically, you know, like I said, it's pretty straightforward for the bifenthrin mitigations, that there's, no applications to be made, not or on the garage interface for any product containing bifenthrin, unless it's somehow protected from both rainfall and sprinklers, which I don't, at that point, I don't see how it's a driveway anymore. It's basically a garage. Yeah, so that's the bifenthrin. And then fipronil, I'm really proud of, because I was part of this effort to try to mitigate fipronil. And this was a big collaboration we had between UC researchers, PCOs, and registrants. We developed several scenarios, application scenarios, where the use pattern was restricted around the structure. And then we had the researchers apply that to different homes, volunteer homes, and they measured the runoff coming off the driveway. Whoops, sorry. And then I don't know if you can, this is a little blurry picture, but this is a picture of them measuring the ant populations directly. So it's a very unique study where they, and this is a sign of efficacy, where they measure how many ants were caught or still alive around after the different application scenarios. And lastly, that we had the PCOs. This was very unique. They had over 2,000 structures through their contracts that they conducted blind tests of restricting the applications, and then just counted the number of monthly callbacks. And so this is a very important piece of the puzzle, because to the companies, to get sign off on them, they really don't want their businesses affected, and callbacks are a really costly and time consuming effort. And so we really wanted to get this information to see if it had a drastic effect on what they were seeing. And luckily, everything went as good as planned, with the most restrictive application scenario, which I'll get to in a second. We saw a drastic reduce in concentration in the driveway. We saw the efficacy of the product remain essentially the same. And like I mentioned, the most important piece was that we didn't see any increase in application or in callbacks between before and after the study for the restricted use applications. So that really helped us get the sign off from the professional organizations. And then we proceeded to develop label restrictions with the US EPA. So this is basically the restrictive applications for fipronil that's on the label, which is great, because the labels are very, front and center. We negotiated having the dilution to a 0.03 finished dilution, and a 6-inch up and 6-inch out perimeter band is allowed. And that really, as you can see, that's the only allowable application on the whole structure, or in the whole landscape, is just that perimeter band of fipronil products. There's no applications to be made to the garage door, driveway, or the sidewalks, again, highlighting that the impervious surfaces are important in our piece of the mitigation strategy. They can apply 4 times per year with 60 days in between. But the most unique, I think, aspect of the restrictions was this no application restrictive period between November 1st and February 28th. So if you remember, I highlighted how important stormwater runoff was to transporting chemicals off the landscape, and this period is approximately corresponds to the storm season in California. So we were really happy to get this on the label. So they should, there shouldn't be any applications during that for at least a third of the year. So that's great. We're, in addition to regulations and label changes, we're also investigating structural mitigation strategies. These are biochar filled socks. We've worked with several researchers and found that they were really, they worked well in agricultural ditch settings. They had proven efficacy at reducing pyrethroid concentrations. So I thought, well, we should try to see if we could get similar results in the urban environment. So we've been placing at a couple locations, placing these biochar filled socks at the outlets of storm drains during. Unfortunately, we can only do it during the low flow dry season conditions because otherwise it just overpowers the socks. And then we want to maximize the flow through the socks and then evaluate the efficacy by collecting samples above and below the sock placement to measure the efficacy. So this is kind of a newer study where we don't have a lot of results yet, but we're keeping an eye on how they're working. Okay, so next I'll be moving on to potential sources. Like I mentioned, part of our group's effort is to not only just measure what's in the waterways, but also to try to track them up to see where they're coming from and what are the potential sources. So this is another busy figure. This is a conceptual model that we've put together representing, it's not, it's as complete as we can right now, but it's always a work in progress as we identify new pathways and new sources. So essentially, we try to identify the important product types that are applied as specific application sites. And I've talked a lot about the structural and landscape applications, and then there's some indoor applications as well, and then certain activities that cause a pathway between the application site and move it from the application site into either the waste stream or urban runoff, and then ultimately could be found in our surface waters. So up until now in this presentation, I've been really highlighting a lot of our the urban monitoring, which clearly paints a picture that outdoor use products are a huge contributor to pesticides to urban runoff and into our surface waters, using from the structural landscape applications through storm and irrigation runoff. So that I feel is pretty well set and a proven pathway. So now I'm also going to be just talking a little bit about the potential for pesticides movement into the waste stream. And I'm going to highlight right now that the slides that I'll be demonstrating or talking about today are only concentrating on what's entering the waste stream, or because I'm talking about sources. We're not talking, this is no way indicative of what we're finding in the effluent or found in surface waters, but we wanted to start at least painting a picture and getting a clearer understanding of what is going into the waste stream and what are the important sources. So the first study we did was a wash-off study for topical pet products. Fipronil, imidacloprid, and permethrin are the most common pesticides found on spot-on flea products. And we got volunteer pets that were already using a fipronil spot-on product to apply and then wash. We washed the dogs. It was actually a really fun project. Either 2, 7, or 28 days post-application. And then we collected the rinsate and measured all the fiprils in every in the samples. And what it was very interesting study, even though the products on the labels will say it's it doesn't wash off, and what was it called? I forgot the wording, but yeah, it's not supposed to wash off the dog. We found fipronil, at least detectable levels of fipronil in every single sample that we collected, even up through 28 days after application. And not only that, dogs washed with within 28 days of application could account for 100% of the observed fipronil concentrations in wastewater. So it really painted a picture that this was a definite source and a definite pathway of at least fipronil into the waste stream. Oh, and I should just mention that the next 3 slides, they all have these red boxes at the bottom, and these are all represent the manuscripts that the results are, in case anybody would like to follow up and read more about it. The second study was a collaboration with UC Riverside again, where we wanted to look at foggers, because they, we believe that they have the potential for releasing a lot of a mass of pesticides, and cypermethrin is the most common pesticide in total release foggers. So they took fogger inside a enclosed space, and then they measured the dispersion of the cypermethrin throughout a room, and they could measure it pretty much on every surface of the room. They had detectable levels of cypermethrin, and also they wanted to measure the wash-off potential on different surfaces. And so they did find a big variance between the types of surfaces, but I will say that all showed the potential to be washed off and then therefore transported down the drain. So we know also that, foggers, this does indicate that foggers have the potential for contributing to down the drain. And then sort of kind of putting it a more direct spin on it, we had a great opportunity to work with one of the facilities, wastewater facilities, and to collect single source samples in the wastewater stream, and those included sites that received, I guess we could go effluent from pet groomer, a laundry, professional laundry, and a pest control operator. And then we compared the concentrations to the main laterals to estimate contributions. So in each of these, I'll point out that what the maximum of the lateral, the main line, the concentrations are in this dotted line right here. And you can see the groomer is in red, the laundry is in blue, and the PCO or the pest control operator is in green. And it pretty much jumps out right away that there's really high elevated concentrations of fipronil, imidacloprid, and permethrin at the groomer site, which, if you remember, is the main active ingredients in topical pet products. So we know that it's being washed off at great amounts, and you can see, especially for fipronil and imidacloprid, the median concentrations are orders of magnitude greater than the maximum lateral. So this is really highlighting the potential contribution of that one specific product to the waste stream. And then over here, just to go back to the fogger, we have cypermethrin is elevated, not for the groomers, but for the professional laundry. And as you remember, that's the ingredient in the foggers. So it's highly likely that the clothes and everything that people are picking up in their own households from the use of these product types, and then being subsequently washed at the laundromat are coming off, being washed down the drain, and that's why we're seeing such high elevated concentrations at this site. And I believe that is the end of my presentation, and it's 42 minutes, so I'm surprised I did that well myself.
Yeah. Yeah. Thanks, Rob. Great on the timing and fantastic on the content. It was really awesome presentation. I think what we'll do is we'll switch over to hear from Autumn, and then we'll open it up to Q&A from folks across both of you. But I will just mention, Rob, was the term you were looking for rain fast?
Rain fast?
Yeah, with like that the product would stay applied to your pet and not get washed off.
Oh, maybe. There's a direct link on the label itself. It says, and I'm gonna have to go back and look what it says, but it says it basically cannot be washed off, or it shouldn't be washed off.
Yeah, we had a comment in the chat on that
Yeah.
one.
Oh, okay.
Yeah, great. Thank you.
Welcome.
Autumn, we're ready to move to your presentation for us.
Sounds good. Good morning, everyone. Can everyone hear me?
Hear you great.
Cool. Okay. So I am going to shift gears a bit and talk about what the Wastewater Enterprise of the SFPUC monitors in relation to pesticides. So I don't think that I need to say this, but in case it's helpful for anyone, the San Francisco Public Utilities Commission provides drinking water, power, and wastewater services to residents and businesses in San Francisco and some other small pockets throughout the Bay Area, but mainly San Francisco. So there are 3 wastewater treatment plants within San Francisco. Any wastewater treatment plant in the US needs to have a National Pollutant Discharge Elimination System permit. And the National Pollutant Discharge Elimination System program it was founded under the Clean Water Act and is managed by the EPA. So for the 3 wastewater treatment plants in San Francisco, we have these NPDES permits. And within those permits, it details what pollutants need to be monitored for and the wastewater influent, effluent, and biosolids. And these pollutants that are included in the NPDES permits are largely identified from the California Toxics Rule. Not all of them are, but the majority of them are. And all of the pesticides that the Wastewater Enterprise monitors for are from the California Toxics Rule. And in case this is also helpful information, the California Toxics Rule is it basically sets the water quality standards for 126 priority pollutants in California, and they're a subset from what's regulated federally, but they're very specific to California. So in terms of pesticides that the Wastewater Enterprise monitors for, this is the entire list that we monitor for. These are mainly legacy pollutants, and most of them have been phased out. So not surprisingly, none of these are really in use and definitely are not on the reduced risk pesticide list. And what the results from 2019 till now have shown is that we didn't find any of those pesticides in our influent or effluent or biosolids. Basically, what this non-detect means is that it's just below any regulatory thresholds, or they were at such low levels they didn't even show up. Then we report on these results to annual regulatory reports that we need to submit to the Water Board. So we do look at pesticides that are not priority pollutants, that basically a pesticide that doesn't have a regulatory driver for Wastewater Enterprise, but is something that we are concerned with for a number of reasons. We can still monitor for those, but we do that in collaboration. So mainly we work with the San Francisco Estuary Institute on that. We also do work with DPR. Rob and I have participated in some projects together. And then we also work to less monitoring, but more kind of regulatory side. We'll work with the Bay Area Clean Water Agency's Pesticides Work Group, which is a subset of the Bay Area Pollution Prevention Group. And we have developed a number of programs and projects through that group, but currently the biggest one is fipronil and imidacloprid, which Rob has spoken about, and that's pet spot-on treatments for flea and ticks. And we have developed a bunch of outreach messaging related to what you can use instead of that, or how you can use it in a way that makes it less bioavailable, basically. And I think that is it from me.